Thank you for the introduction. I'm delighted to be able to present this today as a representative from the Pharma MIDD Working Group. Most of the key material I present comes from the ongoing conversations from within this ICH MIDD discussion group. The principles of MIDD stems from the underlying need to provide answers to questions that cannot be addressed, or at least not fully addressed, by just looking at observed data. Modelling simulation is in general used to guide and steer and inform our future world, whether it be in predicting how best to support our growing population, guiding space exploration, dealing with the current terrible pandemic, or assessing the impacts of climate change. Of course, it is clear that no mathematical model can be a perfect representation of the real world, but many can be useful in helping characterize the current situation and assessing future scenarios. In this context, MIDD concerns the practice of using modelling simulation to make drug development more efficient. In this presentation today, I'll cover the following learning objectives. I'd like to inform you what modelling informed drug development is, why it's important for efficient drug development, why there's a need for global harmonisation in this area, and therefore the remit of the ICH MIDD discussion group. The current working definition of MIDD from the ICH MIDD working group describes it as the integration of data from multiple sources in the form of mathematical and statistical models, and the application of these models using simulation to inform drug development and registration strategies to optimize the design of future clinical studies and to address those individualization questions. I'll now go into the coloured text in a little bit more detail. Given this is about making drug development more efficient, a clear focus is using available clinical trial data for candidate of interest along with data from trials and of other drugs used to treat the same disease. Data of interest in model development will normally include all dosing information, available exposure and PK data, biomarker responses, primary and secondary endpoints, and key safety information, along with other covariates such as demographics and genotyping, phenotyping, which may help better characterize individual responses. The point of the term multiple sources is that enrichment can come via analysis across clinical trials, but also data from non-clinical experiments can inform the mechanism of action, a causal pathway, provide relative potency estimates, an understanding of selectivity, rates of onset and offset of pharmacology, all of which can help enable both dose and temporal predictions and extrapolations. The real-world evidence in turn enhances the understanding of the linkage between clinical trial outcomes and effectiveness in the patient population. In terms of mathematical statistical models, these include all empirical, semi and mechanistic models that utilize assumption based upon the understanding of pharmacology, PK/PD linkage, impact of demographics such as age, weight, genotype and pharmacology on pharmacology and disease processes. While there is a focus on the more mechanistic clinical pharmacology principle models, there is no implied exclusion with respect to simpler statistical models which might adequately relate exposure to outcome and be equally useful. In terms of application with respect to drug development and regulatory strategies, simulation from developed models can be used to inform the future viability of an experimental agent early in development, so that candidates unlikely to have an acceptable benefit-risk profile can be terminated early, reducing unnecessary future patient exposure. Simulations can also be used to assess the probability of trial success, and this concept can be extended to assess alternative drug development strategies. And through subsequent interactions with regulatory agencies, it's possible to use these outcomes to align on programs with a higher probability of regulatory success. Application of MIDD also includes optimization of individual trial designs in terms of choice of dose regimen, number of trial arms, endpoints, observation times, population and trial duration, where a focus of this optimization may be to increase the subsequent predictive value of the knowledge gained from the trial in question. Currently, though, the most common application of MIDD is to optimize the dose-dose regimen. This is based upon establishing through the modeling the impact of underlying disease, demographic characteristics and concomitant medication on both the efficacy and safety via sometimes the impact on the pharmacokinetics. And using this knowledge to maximize the benefit risk for subgroups of patients by assessing gain from alternative dosing paradigms. In this regard, simulations can also be used to determine the value of titration based upon outcome at the individual patient level. These outputs can ultimately be used to inform labeling recommendations and the risk management plan. So MIDD has broad application across the entire drug development continuum. In this 2016 FPR paper, over 100 case studies were characterized in terms of drug development phase, drug development question, MIDD approach used, and impact on decision making. This highlights this breadth of application. The green color shows the spread of these case studies across phase and across question, where the questions run from the characterization of the target and candidate early in development through to study design optimization across all of development, through to prediction and characterization of the impact of intrinsic and extrinsic factors on the PK and PD, the benefit risk assessment from early to late, and dose schedule selection through to the eventual label recommendations, but also with respect to differentiation from standard of care and comparators, which may influence commercialization strategies, and finally population selection early, but definitely bridging between populations from sort of early to late development, and this would particularly apply to extrapolation from adults to children. So in terms of the role of MIDD in regulatory review, this summary statement from our ICH MID discussion group highlights that regulatory agencies do in general currently expect to receive and do receive MIDD as part of the submission dossiers. And indeed, many of the agencies, FDA, EMA, PMDA, and MMPA, have published over the years on their experiences in terms of the utility of MIDD conducted by industry or alongside regulatory agencies themselves in enabling study design, therapeutic optimization, efficient bridging across formulation and populations. In some instances, based upon the integration of available evidence and the subsequent simulation using MIDD approaches, it's been possible to waive the need for further studies. And this is where we have an adequately qualified model, and the context of use means that the benefit to the patient for using MIDD evidence outweighs that of having to run additional trials. One publication of particular note is this 2019 paper from the FDA ClinPharm Pharmacometric colleagues, which summarises their view of the current practice and future considerations. This paper highlights the application of MIDD across areas of dose optimization, clinical trial design, and where MIDD has given support for evidence of efficacy. Interestingly, there is also situations where MIDD has informed regulatory policy. Examples of the latter would include the use of concentration QT analysis as an efficient alternative to running thorough QT studies as per the updated ICH E14 guideline. Also, these approaches have been used to inform the policy around extrapolation of efficacy from adults to children greater than 4 years of age in the treatment of partial onset seizures. Within this paper, the value of the now ongoing highly successful MIDD pilot program, where companies can work more closely with the FDA colleagues on the MIDD plans, is discussed. The likelihood of future growth in MIDD applications implied along with expected greater use of mechanistic models to increase confidence in extrapolation. Similarly, the potential of machine learning approaches in this area and the greater use of real-world data and real-world evidence is also highlighted. Alongside the increase in regulatory submissions using MIDD, there's been a growing global interest in standardisation in this area, coming both from industry bodies and regulatory agencies. The appearance of these guidelines has provided the substrate and motivation which has led up to the formation of the ICH MIDD discussion group, which I'll now talk more about. So here is the construct of the discussion group. You can see it includes colleagues from across the globe, from industry and regulatory bodies, and we've now been meeting since January. I've had 11 meetings discussing this area and how we can move forward at the ICH level in terms of developing guidance which would further help harmonise and improve the quality and application of MIDD to drug development. So the remit of this discussion group is that it would finalise the scope for a general principles guideline on MIDD and position this proposal with respect to update of guidelines that are MIDD focused at the moment, such as the ICH E4 guideline on dose exposure response, and also develop a multi-year plan for the integration of MIDD into existing ICH guidelines while potentially consider developing new guidelines for particular applications such as POP-PK, PBPK, etc. The remit of this group is 1 year in duration, and we're looking to read out at the end of this year. So clearly, if we don't harmonise, we'll miss opportunities to fully leverage MIDD. We'll continue to see situations where MIDD application is accepted in one region but not accepted in another region. As a result, we'll see the over-reliance on observed data to answer drug development questions and regulatory questions, leading to inefficient drug development. Strategies to develop new medicines will not gain from potential efficiencies, and clinical studies may not provide the necessary learning opportunities in terms of exploring the dose exposure response surface. Finally, development could take longer and patients will need to wait longer for innovative therapies to be available. As part of our discussions, we've debated what are the biggest challenges to further implementation and application of MIDD. Here are 5 possible reasons. I'll run through these and give you a moment to consider yourselves before providing what we aligned on as a discussion group. So option A is that there is limited opportunity to apply MIDD in drug development, and that is the biggest challenge. Option B is a lack of belief that MIDD can be useful in drug development. Option C, there's a lack of common understanding between technical and non-technical experts. Option D, there's a lack of common standards and understanding of terminology. And option E is that there's a variable level of integration of MIDD into regulatory submissions. So I'll pause for a moment and let you consider these options and then show you what we consider to be the biggest challenges. Yes, indeed, as emphasised in the presentation, option A is not an issue. We are seeing a significant growth in application and opportunity for application year on year, both within industry and the regulatory setting. Option B, I think it's clear that, like in many other industries, modelling simulation, and in our case, MIDD, definitely has a role to play in making drug development more efficient. And we also believe that this is a commonly held view across global drug developers and regulators. However, we do think that option C may be one of the biggest challenges. From surveys conducted and shared experiences we've discussed, it does appear that aligning on what MIDD is, when and how it can be appropriately applied, provision of confidence in its evaluation within a regulatory setting is a critical step in further implementation. We believe that gaining alignment on the assumptions utilised within a particular MIDD application may be one of the ways of advancing the common understanding. There's also a need to work on common standards and gain alignment on terminology used across regions and types of applications. One of the aims of the General Principles Guideline will be to provide a general framework in this regard. With respect to option E, we are aligned on the need for documentation standards to improve how MIDD is presented within global submissions. In summary, MIDD can optimize drug development resources for industry. It can also aid regulators in containing unnecessary human exposure and enhancing regulatory review. For patients, it can reduce healthcare risks by limiting exposure to investigational drugs and perhaps providing earlier access to breakthrough medicines. The MIDD discussion group is aligned on there being a need and measurable gain from developing a general principles guideline, and an update ICH MIDD topic proposal with this in mind is currently under development. I'd like to thank you for your attention and interest.
Hello, so as mentioned, my name is Robyn and I'm the Director of Patient-Focused Drug Development in the Center for Drugs at the US FDA, and I'm happy to be here today to talk with you a little bit about the recently published ICH reflection paper on patient-focused drug development. Today I'm going to provide you with a little background to set the stage for the paper, and then I'll walk you through what we see as opportunities for incorporating patient experience data into medical product development. We'll touch on the possible topics for future ICH guideline development, and then I'll close with an update on the current status of the reflection paper and a brief discussion of our next steps before we move on to a question and answer session. The ICH Patient-Focused Drug Development Reflection Paper, or the PFDD Reflection Paper, sets out to do two things. The first is that it identifies key areas where incorporation of the patient's perspective may improve the quality, relevance, safety, and really the efficiency of drug development and may inform regulatory decision-making. It also presents opportunities for development of new ICH guidelines in order to provide a more harmonized approach to the inclusion of patients' perspective in a way that is methodologically sound and sustainable to both regulated industry and regulatory authorities. And as we start the discussion, I thought it'd be helpful to provide a brief reminder of where we are with the reflection paper. The paper was endorsed by the ICH Assembly in November of last year, and it was open for public consultation through March 7th of this year, of 2021. And that brings us to where we are today. And so I started out kind of talking about the goals of the reflection paper, but now we're going to talk about the why of the reflection paper. And it really starts with the following premise, that patients are experts on what it's like to live with their condition. Patients have direct experience in living with a condition. They have firsthand knowledge of the impact of the condition on their life and how they feel and function. They bring a unique and valuable perspective to drug development, one that cannot be provided by the clinical, scientific, legal, and other experts. It's important for health authorities and for drug developers to incorporate the patient's perspective beginning early in the drug development process. And so as continued advances in communication technologies, internet, social media, and a proliferation of information services and sources, as well as patient advocacy and patient engagement, have created a rich yet really complex environment for eliciting and incorporating patient perspectives through the drug development process, it really has become increasingly critical to develop a harmonized approach to collecting and incorporating patient perspectives into the drug development process. And throughout this process, there's an opportunity to increase the quality of the development program through effective inclusion of patients' perspective. And these opportunities can include understanding the clinical context for medicine's development and evaluation, informing product design features, including formulation and delivery modes that minimize burden and support adherence, development of endpoints that reflect benefits that matter most to patients, and identifying which adverse event endpoints are most important to patients. Designing clinical trials that support better enrollment and retention, and informing regulatory decision-making, including patient acceptability of benefits versus risks versus tolerability concerns, and effective risk management. And of course, we know that in many cases, drug development plans are already including the patient perspective. However, methods for identifying, collecting, and analyzing what is meaningful to patients are not standard, nor are they harmonized. Similarly, systematic studies of patient preference may not be necessary in many clear-cut situations, but when they are, it would be beneficial that the methods follow agreed-upon standards. And so if methodologically sound data collection tools are developed and used within clinical trials, and sound standards for the analysis, reporting, and application of the results are developed and used, patient input can provide a valuable, direct source of evidence regarding the benefits and risks of a drug, including, where needed, relevant information on patient preferences. And to maximize the benefit of patients' perspectives in these areas, regulators and drug sponsors need to employ methods and measures that ensure the information collected is of sufficient reliability, validity, and representativeness to be used as a basis for planning and decision-making, can be deployed in a timely and sustainable way, will be relevant to patients and the caregivers living with the same disease in multiple regions of the world, reflect concepts such as pain, fatigue, physical function that matter, and measure changes that would be meaningful, and account for heterogeneity or subgroups. And this brings me to the next section of my presentation, ways of incorporating patient experience data. And we touched on this above, but we're going to go a little bit deeper because, as we discussed, patient perspectives can be valuable in addressing specific questions to inform development programs and related regulatory decision-making. And so the following are really questions that may be asked during the discovery and developmental phase where patient input is critical. Equally important is how we handle the information we gain from these questions. And so let's start off with asking, what are patients' unmet needs that suggest potential drug targets? Another question, what disease effects and treatment burdens matter most to patients and might be addressed by a medical therapy? Does this value by subpopulation? If so, how? What would be the best way to measure these effects? What endpoints are most relevant to patients, and can these endpoints be incorporated in clinical trials in a manner that will be robust enough for regulatory decision-making? What is a clinically meaningful change in an endpoint from a patient perspective? How should we define meaningful change in a patient over time? Another example of where patient input is important is in the case of patient preference. And with that, we really start to think about some of the methodologic considerations. So in thinking about patient preference, we need to think about what methods and approaches could be used to identify, for example, which treatment benefits would be most desirable to obtain and which risks would be most important to avoid. Or perhaps we want to explore what patients might consider to be acceptable trade-off of an increased expected harm for a specified increase in expected benefit. In the second example, we think about what are the relevant methodologic considerations when a sponsor conducts patient preference studies to provide credible and reliable findings to support regulatory decision-making. And really, this leads nicely into a discussion of future guideline development. And really, the reflection paper proposes two possible topics. The first is a new ICH guideline that will discuss what to measure in a clinical trial, including how to refine a set or list of important impacts and concepts identified by patients in order to select, modify, or develop clinical outcome assessments that can demonstrate meaningful change. It might also discuss defining endpoints and determining what constitutes meaningful change. The scope of this guideline would include qualitative and quantitative methods to identify disease or treatment impacts important to patients that would be candidate concepts for measurement with patient-reported outcome measures or other types of clinical outcome assessments, or in quantitative assessments of the patient perspective. The second topic would be a guidance looking at methodologic approaches for eliciting and analyzing patient preferences to inform drug development, benefit risk assessment, and other decisions. And while there are not a lot of details on exactly what these guidelines may look like within the reflection paper, I think that this is a good thing, because if these concepts move forward, it will allow the expert working groups the flexibility to adjust to respond to feedback from stakeholders and meet their needs. Another thing that this paper does is that it opens the door to new or additional opportunities to incorporate the patient's perspectives into existing ICH guidelines, something we're already seeing as part of the revision of both ICH E8 and E6. And moving on now to the final section of my presentation, I'm going to share some updates about possible next steps. So back to the public consultation, as I previously mentioned, the consultation period closed on the 7th of March of this year. We received over 300 comments, and those comments came from more than 35 stakeholders from around the world. These stakeholders represented a broad range of interests. We're in the early stage of reviewing the comments, but our initial impression is that the comments were generally supportive of the PFDD reflection paper and of ICH developing harmonized guidelines on methodologic considerations for including patient perspectives in drug development. Many of the comments contained recommendations to be considered by an expert working group if a new topic proposal for the development of a guideline moves forward. The reflection paper also notes that there are existing regulatory guidances, a number of ongoing collaborative efforts, and a large body of existing literature that will support the development of the proposed guidelines. We recognize that both regulatory authorities and companies have limited staff with the requisite expertise in psychometrics, related statistics, and decision sciences to undertake this work. And so we've recommended that the outlined work be considered to start after a substantial completion of related ongoing work. And so here on this slide, you'll see some limited examples of the ongoing work. Specifically, we call out the work currently being done by IMI Prefer, the Innovative Medicines Initiative, and work being done here at the US FDA as we develop our patient-focused drug development methodologic guidance series. Although we do, of course, acknowledge that this is just a snapshot of the work that is being done in this space. And so now I'd like to just touch a little bit on ICH's goal of transparency. And so sharing information with interested stakeholders and wider public in an accessible and understandable manner is important to ICH and serves as a mechanism to build trust. By making information publicly available, the ICH Assembly and its Management Committee hope to make the ICH process ever more transparent so that all stakeholders can be informed on important decisions and real-time progress of the ICH guideline development. Particularly for work in this area, we understand the need to address the practical involvement of patient and other external stakeholder organizations throughout the process. And this involvement needs to go kind of both ways. Not only do we want to share information, but we need stakeholder feedback. And so the proposed process included the posting of the reflection paper for public comment. And if the proposed guideline work is advanced in new topic proposals and is subsequently endorsed by the ICH Assembly, the new topic concept paper and business plan will include plans for public consultation and engagement similar to the approach being taken by the ICH E6R1 group and incorporating the learnings and best practices from that group, as just discussed by my colleague, Dr. El-Zarad. And so now, for ease of reference, I've included a link to the PFDD, or Patient-Focused Drug Development Reflection Paper. And I'd like to open the floor to any questions. Thank you so much.
It is right that Dr. Mullen gets the first question. So, Dr. Mullen, has the pandemic impacted the development or quality of ICH guidelines, monographs, etc.? For instance, due to lack of face-to-face meetings by the applicable ICH committee meetings? Any pros and cons?
Thank you, Brenda. The short answer is yes. This harmonized guideline work is, addresses technically complex topics, advanced topics, reconciling factors and considerations, and harmonization is challenging work. And typically, the working groups, have TCONs are part of their mode of operation, but the two face-to-face meetings each year in which the work groups are able to get together for, say, 3 to 5 days. They are typically working 10 to 12 hours a day. It's very intense, but they're in the same location, same time zone, face-to-face, and this helps because, body language can be very important for many of the ICH participants. English is not their first language, and so it's more challenging to do this all virtually, which we've been doing since the start of the public health emergency. So this has naturally created some delays and challenges for the groups. I think we've all gotten very familiar and facile with many virtual platforms. People have gotten probably maximizing the use of these platforms, but it is delaying things. I think most work groups have been delayed in the range of 6 to 12 months, sometimes even a little bit longer, and it does reflect just the challenges that I've just described. So thank you.
Okay, thank you very much, Dr. Mullen. And then the next 3 questions will be directed to Dr. Vesper. So Dr. Vesper, what is the rationale for establishing a PDE in the absence of carcinogenicity or reproductive toxicity data, please?
So thank you for the question. A PDE can be derived considering data, like additional data other than carcinogenicity and reproductive toxicity studies, and can include, for example, repeat dose toxicity studies. And deriving a PDE is very useful as it provides a harmonized value that can be employed by both regulators and the pharmaceutical industry. So that said, the expert working group will only consider deriving a PDE and developing a monograph for a compound that has sufficient data. If there's insufficient data and there are no structurally alerting functional groups, for example, for mutagenicity, then it could be considered as an impurity that can be controlled per ICH Q3A and B.
Thank you very much, Dr. Vesper, for that response. Next question for you. Is there any specific requirement to conduct toxicity studies for calculating PDE? Who determines the PDE? Any specific regulatory agency? And are non-rodent species considered in calculating PDE values?
That's a good question. Thank you. So there are no predetermined set of studies that need to be conducted prior to calculating a PDE. All publicly available study data is considered, which includes non-rodent studies. The data is evaluated by members of the ICH Q3C expert working group, which consists of both regulators and industry representatives across the ICH regions. So there are many country-specific regulatory agencies that are represented.
Thank you, Dr. Vesper. And this is your 3rd question, the last in this cycle of questions for you. How do we know the PDE for the residual solvents that were not listed in ICH Q3C? And what if no toxicity data is available to that particular residual solvent?
So another good question. Thank you. So if there is no PDE listed in the ICH Q3C data, and if there are no toxicity data available for this compound, then the molecule can be controlled per ICH Q3AB. There may be one step that should be considered between that is to determine if there are any structurally alerting functional groups for mutagenicity. And if there are, then the recommendations in ICH M7 should typically be followed. Otherwise, it can be controlled per Q3A and B. Thank you.
Thank you very much, Dr. Vesper. Our next round of questions will go to Dr. El-Zarad. Dr. El-Zarad, this new way of monitoring sites remotely has taken a peak than usual. Will the same be included in ICH E6 guidelines in future?
Thank you, Brenda, and thanks for the question. So, of course, with the caveat that the work on ICH E6 R3 and specifically the details in the annex are ongoing, I can highlight that the EWG, the expert working group, discussed highlighting primarily the purpose of monitoring, and that the purpose can potentially be achieved in different ways and in using different monitoring approaches. This, of course, may include the consideration of remote monitoring or even a hybrid monitoring approach in appropriate circumstances. Also, I would add that the expert working group discussions highlighted the importance of a risk-based approach to monitoring, and that the monitoring approach should be designed, including the monitoring approach and the monitoring plan, should be designed to fit the characteristics of the trial. I hope that's a little bit helpful.
Thank you, Dr. El-Zarad. The second question for you, what is the FDA's position on implementation of machine learning and AI into clinical trial operations, QA, and monitoring?
Thank you for the question. Clearly a very timely topic. So we are very aware of the current and potential uses of AI across therapeutic development spectrum, and I'm including here drugs, biologics, and devices. Clearly, there is an opportunity where AI-based solutions can make therapeutic development more efficient. We are seeing great potential, for example, for AI from early stage drug candidate identification to PK and PD modeling to clinical trials, recruitment, and retention, which is the main topic for today. The Center for Drugs at the FDA established a few months ago the AI Steering Committee, the Artificial Intelligence Steering Committee, as a focal point for us to bring the AI-related activities and effort and try to understand and train the training needs for our staff. So it's very much a subject that we're very keenly aware of, and we're very interested in also making sure that these innovations are facilitated in an appropriate manner. Since today that we're highlighting the work of ICH and E6 EWG, the topic itself came actually up in the discussions of the EWG as well, and we see it as an integral part of the whole digital ecosystem that we have to consider collectively, making sure we take advantage of the opportunity it has for us, while also understanding the caveats and the issues we need to address from a conduct perspective. Thank you.
Thank you, Dr. El-Zarad. This is the last question for you in this cycle. What are the steps being taken to address the administrative burden on site staff considering the increased pace of changing technology in the clinical trial field?
Thank you for the question. This is another really important topic and a key issue that the expert working group is considering across all processes and approaches that are part of the clinical trial conduct. As you can imagine, clinical trials are substantial undertaking, and that will require effort, require focus and thoughtfulness, which, burden is, natural part of that. I think the question is how to eliminate unnecessary complexities that add burden, but have little of value in return. And how to address the needs and requirements of the specific trials, from a point of view of where to focus the efforts, where to focus the majority of the resources, if you would, in a specific area. So it's important to mention that burden should be considered across the trial life cycle, but also across stakeholders. For example, when you're seeing trials increasingly in a community setting or reaching out to patients in their environment, healthcare providers might be a part of that. Of course, participants themselves, have the burden of participation, and understanding how collectively we can design and conduct trials in a way that minimizes complexity for all stakeholders and not just for site staff. I think it's really important, and it's an important topic that the EWG is considering. One of the core elements the EWG is highlighting is a risk-based proportional approach that focuses the effort and attention on what matter most for participant safety and to ensure the reliability of trial results. So that you'll see that as a focus for us as EWG. I will end with a kind of a small example that the EWG discussed that, for example, training requirements shouldn't be a blanket requirement, but it should correspond to the role expected to be played in the trial. I hope that highlights this issue a little bit better. Thank you.
Thank you very much, Dr. El-Zarad. And now we'll move on to Ashley Bohm for the next series of 3 questions. Ashley, could you please comment on the process of implementation of Q12 in FDA? What are the main barriers for this implementation?
Thanks, Brenda. I appreciate the question. So our primary barrier was actually getting the Q12 guideline published as a final FDA guidance. And now that we've done that, we are full speed ahead into implementation. I'm pleased to say that the different tools outlined in Q12 all fit within the US regulatory system. So unlike some of my regulatory counterparts, we did not have to make changes to our regulations to allow for implementation of Q12. Thank you.
Thank you very much, Ashley. Next question for you. How is ICH Q12 and guidance for industry post-approval changes to an approved NDA/AANDA associated?
Thanks, Brenda. So the concept of established conditions, which explains and becomes an agreement between the applicant or the authorization holder and the regulator for which elements of the CMC section of an application have to be reported if changed, is actually a voluntary activity. So in an instance where an applicant chooses not to use established conditions, the guidance here on post-approval changes to an approved ANDA or NDA or any of our existing SUPAC guidances are still applicable to guide applicants in determining the appropriate approach to submission of post-approval changes. Where an applicant or authorization holder chooses to propose established conditions, they may propose established conditions and reporting categories for changes to those established conditions that are different than what's in the SUPAC guidances or the guidance named in this question. And that requires, obviously, scientific justification for why the risk level for that change for this particular product, and given the process and product understanding of the applicant, why that information justifies a lower reporting category. But that's one of the benefits of the established conditions approach, is that in a number of cases, lower reporting categories can be proposed and justified. Thank you.
Much appreciated, Ashley. And the last question for you in this cycle. You mentioned a CDER map on Q12 implementation. What about CBER?
Thank you for that question. So yes, my understanding is our colleagues in the Center for Biologics are developing what they refer to as an SOPP, or standing operating procedures, that will help to guide their assessors as they incorporate and implement Q12. Thanks very much.
Thank you, Ashley. And our next round of questions goes to Dr. Marshall. Dr. Marshall, thank you for a really interesting presentation. Has application of MIDD to date shown any therapeutic areas where this has worked really well? And conversely, are there any therapeutic areas where MIDD has not been such a success?
Thank you for the question, Brenda. Yeah, and thank you for the interesting question. Yes, a very good question. And the bottom line is there's no particular therapeutic area that would benefit or not benefit from the application of MIDD. All areas have been shown to have benefit from application of MIDD, and many of the review articles by industry including the one I presented on, which was the FPA one, as well as FDA and EMA and other regulatory agencies across the globe have shown that the examples are really across all therapeutic areas. But I think to answer the question a different way is there are certain situations where MIDD has greater application, and that is where there's a good understanding of the relationship between dose, concentration, biomarker and effect. And where you have that relationship and it's well established, then I guess you get the best benefit from MIDD. And of course that can happen in any efficacy and safety situation, just depends on that understanding of the mechanism of action and its link to the biomarker of interest and outcome. So thank you for the question.
Thank you very much, Dr. Marshall. And the last question for you in this cycle, can you comment on the advantages of scenarios where the FDA MIDD pilot program meeting has over the existing EOP2A or any other meeting format for that matter? And is it possible to provide insight on industry use of the MIDD pilot meeting now that it's nearing its end? Thank you.
Yeah, again, another very good and topical question. Indeed, the industry bodies have reviewed their use of this very innovative approach from the FDA, and the IQ consortia in particular have just written a paper within Clinical Pharmacology and Therapeutics that summarizes a survey result from across at least 20 companies. And it's a resounding positive that this has been of tremendous value to industry, not only in allowing us to get to questions we could not normally get to within your past procedures like the one raised, but also the end of phase 2 meeting as was, or current procedures. And I think the real gain is that this is technical experts leading the engagement from both sides, where there are the other members of the clinical and regulatory team are there, but they're hearing the technical discussion and I think getting confidence within these applications. And this has really helped the industry get clarity on these questions, use these approaches to reduce drug development time and cost in that they're targeting their resources to the key questions that are required under the MIDD predictions to move this area forward. And the value there is not only in that alignment between the technical experts, but it's really allowing industry to align with their decision makers as well by bringing that clarity back. So in itself, it's done a long way of moving forward MIDD, and I really like to thank the FDA for having rolled out this innovative pilot, and we do really hope that this will go forward into a standard procedure that we can work with in the future, and of course that this is mirrored across the globe. Thank you.
Thank you very much, Dr. Marshall. And our next question goes to Robyn Bent. Question: what are the next steps, and when is now planned the first guideline on PFDD step 1?
Great, thanks so much. That's a great question. And so just as an answer, we are currently reviewing the stakeholder comments that we received. We really want to make sure that they're given appropriate consideration. And as we do that, we'll determine how to best incorporate the feedback as we move forward. It's possible that a revised reflection paper could be issued. Since the feedback we received does appear so far to be pretty supportive, as I talked about a little in my talk, we're still working out the specific timeline for any guideline development. I think there's a lot of factors that will go into that, and I don't think we can provide specific dates at this point. Thank you.
Thank you very much, Robyn, for that. And our next questions, we start back with Dr. Vesper. Dr. Vesper, thank you for the valuable presentation. Regarding TBA, PDE is derived considering lifetime exposure. If a drug exposure is up to 30 days, can factor F3 reduce accordingly?
Thank you for that great question. So factor F3 is a variable factor defined in the ICH-Q3C guideline to account for toxicity due to short-term exposure related to the animal toxicity study and not duration of exposure in humans. So for example, if the PDE is derived based on a 6-month study in rodents, a factor of 2 is used. Whereas if a 3-month study is used, then a factor of 5 is applied. So it's not related to duration of exposure in humans. Thank you.
Thank you, Dr. Vesper. Next question for you. When, which month that is, will the draft addendum to ICH S1BR1 be available to the public for comment?
Thanks for that question. So the draft addendum was published on the ICH website on May 10, and it is currently available for review. At this time, regulatory agencies are in the process of launching their consultations. So unfortunately, I don't have specific dates for you, but can only state that comments can be submitted to the regulatory agencies sometime in the near future. Thank you.
Next question for you, Dr. Vesper, is a little bit longer, so bear with me. Could you please explain why, in cases where a WOE assessment indicates that a 2-year rat study will not add value to the assessment of human carcinogenic risk in humans, mouse carcinogenicity studies, the 2-year study and/or short-term transgenic mouse study are not recommended in the EU, but will remain the main approach in other ICH regions? Isn't the purpose of ICH guidelines to present harmonized testing requirements that are accepted across all ICH regions with a view to avoiding unnecessary testing?
That's a really great question, and one that was extensively debated at the ICH table. So for most regulators in the ICH S1 expert working group, it was agreed that a mouse study should still be part of the carcinogenicity testing plan, as the prospective evaluation study did not evaluate the value of the mouse study, and rat study results cannot be directly extrapolated to mouse study results. Furthermore, there's an option to conduct a 6-month transgenic mouse study and not a 2-year mouse study. So this was the opinion that was this opinion was not shared by our EMA colleagues, and it was clear that in order to be able to reach consensus on the document as a whole, it was necessary to include this statement. Thank you.
Thank you, Dr. Vesper. Our next couple questions will be directed to Dr. El-Zarad. Question: will you be kind enough to define stakeholder in the context of all that you discussed?
Thank you for the question. I know I mentioned stakeholders in a different context, so thank you for the opportunity to explain. I mentioned initially the term stakeholders in a sense of those involved in the clinical trial, and in light of, again, the increased use of technology and in light of potentially clinical trials getting embedded in healthcare settings, you can imagine how those who are involved in a clinical trial, they're going to be a diverse, increasingly diverse groups. And that's what I meant by looking at the larger umbrella of stakeholders who could be part of the clinical trial. And the most obvious stakeholder, of course, are the participants in that case. So we should consider the totality of stakeholders who are involved. Now, I mentioned also stakeholders in a sense of our engagement efforts. We realize, for example, that we would benefit from engagement with stakeholders, specifically those from academic research background. And I've showed the list our current stakeholder representatives. We call them the stakeholder representatives as they represent groups and constituents, from the research backgrounds. And we found that engagement to be really helpful because we were able to bounce certain language, with them and try to see how we can minimize potential multiple interpretation. So in that sense, we have those stakeholders groups that I mentioned, those who are involved in a clinical trial as large, but also I mentioned stakeholders in the context of our engagement efforts. I hope that helps a little bit.
Thank you, Dr. El-Serraj. Your answers are always very helpful. Next question for you, the last one in this round. Would GCP E6R3 be applicable to medical device trials? Thanking you in advance.
Thanks, Brenda. The answer is no. The E6R3 is focused on drugs and biologics. As you can imagine, device trials may take a little bit of a different context, may have different sets of requirements or approaches, although there are clearly some overlap. So the answer is no. This is specific to drugs and biologics in interventional clinical trials. Thank you.
Thank you for that very clear answer. And next question goes to Ashley. Ashley, when do you expect Q12 training materials to be available to the public?
Thank you for the question, Brenda. So we have actually divided the Q12 training materials into two parts. The first part is a set of slides that will cover the material in the guideline. And we have submitted those training materials for adoption by the Management Committee and Assembly. And once adopted, those will be posted to the website. So I'm hoping in the next month or so, we'll see those available. We are continuing to work on the case studies. And so we expect that to take a little bit more time to do because they are longer and more involved with narrative text. Those are a little slower to be able to be developed using teleconference and not being able to be in the same room. But we do hope to have those available later on this year. Thank you.
Much appreciated, Ashley. Next question, and we go back to Dr. Vespa. When will these ICH proposed limits apply to these three residual solvents?
Thank you for that question. So the monographs and PDEs have reached step 4 of the ICH process and are considered final. And now the revised version of the Q3C guideline is in the implementation phase within the regulatory regions. So yeah, just to reiterate, these are final values.
Okay, thank you very much. We have 9 minutes left on the clock, and Dr. Vespa, this next question for you is a multi-question and may take us up to the top of the hour. So I think I'll attack it one question at a time. So here we go. Newer anti-epileptic drugs, AEDs, launched in the last 20 years, appear to be safe as no carcinogenicity has been demonstrated either during regulatory testing or post-market surveillance. Here's the first question. Does the ICH S1B R1 Addendum address the need for conducting the 2-year rat carcinogenicity study and the 6-month mouse carcinogenicity for new molecules being developed in this class of drugs or in such classes where recent evidence has shown no carcinogenic potential?
Great, thanks for the question. So the ICH S1B R1 Addendum does not specifically discuss this class of drugs. Rather, it discusses the type of data that goes into a carcinogenicity weight of evidence assessment, which includes data on other compounds in the drug class. Regarding the second part of the question, The ICH S1B R1 addendum is only in step 3 of the ICH process, which means it is in draft form and is not finalized. For this reason, it can't be considered that the addendum is in effect, and I would encourage you to approach specific regulatory agencies for compound-specific advice. Thank you.
Thank you, Dr. Vesper. Second part of the question. Given that this addendum will be finalized only in 2022, I think maybe you have already answered this, but let me continue. What do you recommend sponsors do if they have already begun the 2-year rat carcinogenicity study? Should they simply continue to follow ICH S1B R1 guidelines in option 1 and conduct a 6-month rat carcinogenicity study? Or does ICH recommend they pause and encourage a dialogue with ICH regulatory agencies such as FDA regarding the need for further conduct on the 6-month rat carcinogenicity study?
Yes, so I read too far ahead in that document. So yes, I have already answered the question, and just to briefly reiterate that it's only in step 3 of the ICH process, so this can't be considered a final document. And if there's any compound-specific questions at this point in time, since the addendum is not finalized, I would encourage a discussion with the specific regulatory agency.
Thank you, Dr. Vesper. My apologies for the duplication of question there. Let's go to Dr. El-Zarad. We have 5 minutes. Dr. El-Zarad, I understand that the Wellcome Trust is working on a good clinical practice initiative. How does this effort compare or contrast with ICH's? Have you been in consultation with the organizers of this effort?
Thank you for your question. We are aware of the efforts of the Wellcome Trust. I believe it's called the clinical trial, the Good Clinical Trial Collaborative. And my personal engagement was in a form of being on as an advisor, participating in a couple of their meetings. So we are aware of the work. We also have Dr. Martin Landry, who's also involved, I believe, as part of the leadership of the Wellcome Trust initiative. He's one of the stakeholder representatives that help us within the work that we're doing right now on E6R3. Now, as for the relationship between the guideline, I don't think there is a direct relationship. ICH E6, as you can imagine, is the regulatory guideline, is the unique regulatory guideline for clinical trials that are intended to support regulatory submissions. I think generally we all share the interests of making sure clinical trials are efficient, clinical trials are conducted in an appropriate and ethical manner. So that's the hope, is that we're aligned with Wellcome and with the rest of the community at large on those key goals. So I hope I'm touching on the question in a way that answers it. I would encourage you to participate in the web conference that we have next week. That may also shed a little bit more light on our work in general and the scope of ICH E6 and hopefully the work of the EWG at large. So thank you very much.
Thank you, Dr. El-Zarad. That gives us the end of our Q&A session, but I wanted to say, if anyone didn't get the message Dr. El-Zarad was sending, please attend the meeting next week. Okay, thank you all.